Hello, everyone, and welcome. We'll just wait a couple of minutes to let everyone join. Good morning, good evening, good afternoon to all of you. It's 1 o'clock in Geneva, so we'll start now. I'll start by welcoming all of you to today's EPIWIN webinar, which is the final EPIWIN webinar for 2024. A special welcome to those of you who are joining from very early or very late in the evening and from far away. Thank you so much. I'm Supriya Bezbara. I'm the lead for EPIWIN in WHO. For those of you who are not familiar with EPIWIN, it stands for the WHO Information Network for Epidemics. And our goal is to make the science behind health emergencies accessible, understandable, and actionable for all so that it becomes useful and with the aim of having an impact on the ground in everyone's lives. Today, we have a very exciting topic. It's on pathogen genomics for health emergencies, spotlight cholera. And we have a very eminent speakers panel. For more, without any further ado, I will hand over to the person leading this, Timothy Dahlman, who's from WHO HQ Berlin Hub and is an advisor for genomic surveillance. But before I go there, the usual housekeeping rules. Those of you who are familiar with EPIWIN, who are part of the family, will know this already. We'd love to hear from you. EPIWIN is all about being part of the family. So please introduce yourself in the chat. Tell us where you are, where you come from, what you do. We love to hear from you. But if you have questions, as you know well by now, please put it in the Q&A box, not in the chat, because that's where we'll see the questions. Sometimes we can miss it in the chat. We welcome your comments and thoughts and observations, but please keep it relevant and to the topic. Please also keep it positive and constructive. We really do not like anything negative or that's rude or negative. So please keep it polite, constructive, positive. We're here to learn from each other. This webinar is being recorded and the recording will be shared. It'll be shared on our website and also by email to everyone that's registered. We also get a lot of questions about certificates. So we want to let you know that this is a knowledge sharing platform. It's about sharing the latest knowledge. It's not a training. So we do not give certificates to everyone. However, if you request it and complete the post-webinar survey, and in the survey, also explain how you're going to use this information extensively, not just a one-line answer, then we will, at your request, share a certificate with you. Now, without any further ado, welcome once again, and I hand over to Timothy Dalman, Dr. Timothy Dalman, to introduce our very eminent speakers and take over the moderation from here. Over to you, Timothy.
Thank you, Supriya, and thank you everyone for joining. This special edition of Pathogen Genomics and Health Emergency webinar series, co-hosted by the EPIWIN, the WHO Cholera team, and the International Pathogen Surveillance Network. So a little bit of background on IPSN. So the International Pathogen Surveillance Network is a global network of genomic actors brought together by the WHO Hub for Pandemic and Epidemic Intelligence in Berlin. Its vision is a world where every country has equitable access to sustained capacity for genomic sequencing and analytics as part of its public health surveillance system. And it functions to create a mutually supportive network that amplifies the work of its members to accelerate progress in pathogen genomics, improve access and equity, and ultimately contribute to improved public health decision-making. To inform country capacity building activities, this webinar series aims to provide a forum for expert and evidence-based discussion on the role of genomics in response to ongoing outbreaks, considering where and where not the use of genomics can assist in public health decision-making. So this is the second webinar of the series. And really, this is around informing country capacity building activities. This is the second webinar of the series, and we spotlight cholera, whose global resurgence was classified as a grade 3 emergency by the WHO in January 2023. And today we'll discuss the use of genomics in expanding our understanding of regional and global cholera transmission dynamics and its role in strengthening the overall surveillance for public health interventions. With that, I'd like to introduce our first speaker, Professor François-Xavier Vail from Institut Pasteur in France. He heads the Enteric Bacterial Pathogens Unit, the French National Reference Center for E. coli, Shigella, and Salmonella, and for Vibrios, including Vibrio cholera. In 2017, he transitioned the laboratory surveillance methodology to whole genome sequencing. He has initiated and led several international microbial genomic studies to better understand the population structure, evolution, and spread of various enteric bacterial pathogens, and in particular, Vibrio cholera. And with that, Francois, I give the floor to you.
Thank you, Tim. Can you hear me? Can we start the presentation?
Yeah, we can hear.
Okay. Good morning, everyone. Thank you for giving me the opportunity to talk about the contribution of microbial genomics to the study of the cholera agent. So let's start with a quick reminder on the disease. Next slide, please. So this is an infection caused by a bacterium of the Vibrio cholerae species and belonging to serogroup O1 and producing the cholera toxin. And this is this toxin that is responsible for the clinical symptoms dominated by an acute watery diarrhea and with a rapid loss of fluids that can lead to severe dehydration within hours with a high case fatality rate if the patient is not treated. So the first priority for the treatment is rehydration, and often antibiotics are given as an adjunct therapy. The transmission of the disease is between humans through direct contacts like body fluids and washed hands, or indirect contacts by water or food contaminated by fecal pollution. Cholera often appears as explosive outbreaks in a particular context, often like war, civil unrest, climatic events leading to human gatherings in camps without clean water, decent sanitation. And if the bacteria is brought in these camps, then the cholera can easily spread. Next slide, please. So currently we are in the 7th pandemic of cholera. which began in an island of Sulawesi in Indonesia in 1961. And it was quite a new bacterium compared with the previous pandemics. And this new bacterium was named biotype L-Tor. It was possible to differentiate L-Tor from the classical biotype by a few phenotypic tests, one being resistance to polymyxin B. So from Indonesia, then the disease spread to Eastern Asia, and South Asia, reaching India in 1964, Africa in 1970, Latin America with Peru first in 91, and finally Caribbean with Haiti in 2010. Next slide, please. So currently the pandemic is very active, and in 22, the number of reported cases to the World Health Organization more than doubled compared with 21, with new countries affected like Lebanon, Syria, and also many large outbreaks. More than 10,000 cases were seen in 10 countries. And also, there was a resurgence of the outbreak in Haiti and Yemen. So it's still in the news, cholera. Next, please. So in the lab, I am Guy from the lab. So what we could do in the past, we had several phenotypic tests. Then during the 90s, we got access to molecular methods like ribotyping, pulse field electrophoresis, and then we were able to sequence single genes like the cholera toxin B subunit genes, the TCPA. But all these methods led to confusion because different authors use different methods, and also all these methods didn't get you the possibility to assess the true genetic relationships between isolates. And then we entered into a new era, the genomic era, in 2000 with the first genomic sequence published of the cholera agent. It was strain N16961, and with two different circular chromosomes. So it was the beginning of a new adventure. But next slide, please. We had to wait for the availability of short read sequencing to be able to play with many isolates at the same time. So by short read sequencing, you can identify different mutations in the DNA, what we call single nucleotide variants. And then with these variants, SNPs, you can make trees. We like trees. So with these trees, you can assess the true phylogenetic relationships between isolates. And if you have a representative collection of isolates from different countries, different time frame, then you are able to infer a spatio-temporal spread of the bacterium. For example, you see AFR12. On the ancestral branch, you have strains from South Asia, and then all the descendant isolate are located within Africa. So it means that there was an introduction from South Asia into Africa. By short read sequencing, you can also get access to assemblies, and with these assemblies, you can study the evolution of virulence and antimicrobial resistance determinants. Next, please. So first point is genomics is the perfect tool to identify the current cholera pandemic agent. Next slide. With this phylogenetic tree, based on more than 250 Vibrio cholerae genomes, you have different clusters, but only two of these clusters or lineage are associated with pandemic cholera. The first one is classical. It was responsible for the 6 first pandemics. It's now extinct. except in for one vaccine strain. The second lineage associated with pandemic cholera is the seventh pandemic Eltor lineage, the current lineage. And you can see they can be differentiated by more than 20,000 SNPs. Next slide. All the other lineage, even if they do contain the cholera toxin gene, do not cause pandemic cholera, but just sporadic cholera that looking like cholera, or small seafood-borne outbreak, but without any secondary case, mean no inter-human transmission. Next slide, please. So it's not necessary to perform a very deep phylogenomic analysis to assign your genome to the seventh pandemic Eltor lineage or other. You can just use MLST with 7 genes to assign your isolate to the true pandemic lineage. You can use this scheme developed by Octavia et al. This scheme is implemented in the Center for Genomic Epidemiology. So it's a web tool, very easy to work with. And if you get sequence type 69, and for some African strain from the Great Lake 515, then this is the seventh pandemic lineage. All other are not associated with the current seventh pandemic lineage. So that's a tip you can use easily. Next slide. Second point, genomics is a perfect tool to monitor the spread of the cholera agent. Next slide. So the first big study dealing with that was published 13 years ago by the Sanger Institute in Cambridge with many collaborators. And so at that time, they were able to study more than 100 seventh pandemic genomes. And they make this phylogenetic tree, and they found out that all the genetic diversity of the bacterium was seen only in South Asia, in countries belonging to the Bay of Bengal. And with this phylogenetic tree, they also saw that there was global expansion from there called wave 1, the oldest, wave 3, the most recent, and also looking at the location of the isolate, they were able to show that there was global, several intercontinental transmission events. And for example, the one in Peru in 91, the bug was coming from West Africa, or the outbreak in Haiti in 2010, the bacterium was coming from South Asia. So that was the first study to answer the question of the spread. So, second slide, next slide, please. So then some studies were applied to a particular continent, for example, Africa. So what we knew about cholera in Africa, it began at the same time during the summer of 70 in 2 different locations, West Africa in Guinea and also in North Africa. Later, cholera was seen in November in the Horn of Africa, and even later in 71 in Angola. It was quite easy at the beginning to follow the disease because it was a new disease at that time. But after several years, the laboratory methods were not discriminatory enough to understand the spread. And the question was, has cholera remained entrenched in Africa since 1970? Next slide, please. So to answer that, we did a huge genomic study with more than 1,000 genomes, including 600 from Africa, collected from 1970 to 2014 at that time, from 45 countries. And when you look at the phylogenetic tree, you can see the color of the tips are colored are according to the country or the continent of isolation. In brown, that's Africa. And you can see different introduction of the bug from South Asia to Africa. For example, T9, the 9 events of introduction, you can see that the ancestral branch are colored in green, and then all the descendants are in brown. So it's the same with T10. So next slide. So it means that cholera has been persisting in Africa, but it was not the same sublineage. Actually, we discovered that there were at least 11 introductions, now it's more than 14, from South Asia into Africa every 6 years in two separate geographies, separated foci, West Africa versus East Africa. And during the first introductions, the Middle East was acting as a springboard, but it's no longer the case. And once introduced, the sublineage could circulate from a few years to up to 28 years, then died out and were replaced by new sublineage from South Asia. Next slide, please. Last point for which genomics is a very good tool is to study antimicrobial resistance for the cholera agent. Next, please. When you look at this case, it's a circular phylogenetic tree in the inner part of the figure, and you have the wave 1, the oldest isolate at the top, and then wave 2 and wave 3. Each lane corresponds to an antibiotic, and the color red means resistance. So, and it's quite obvious that over time, from the oldest wave 1 to wave 3, that you have more and more red color. So it means that accumulation of antimicrobial resistance over time. And even some figures in Africa before 1984, 96% of the isolate were pan-susceptible. But after 2000, none were pan-susceptible, and there was even 6 antibiotic resistant genes per isolate on average. Next, please. So also genomics is very important to study the mechanism of antimicrobial resistance. So the same circular tree, and in this case, it's more the mechanism of resistance that are indicated. And actually, there were 2 phases for antimicrobial resistance in the cholera agent. First, the early stage was with large plasmids of the C incompatibility group. But after the 80s, we saw more and more chromosomal determinants, could be genomic island, like the S60 element, it's a 100 kb element, and with 5 variants, but one is quite prevalent now, it's called ICE VCH in 5, and you can see that all wave 3 isolate contain this agent, this element. We have been seeing more and more point mutation in some genes like GRA and PARC, encoding resistant to quinolone, or reductase NFS A and B for resistant to nitrofurans. Next, please. So what can you do quite easily in your lab is it's quite easy to determine the MR gene content, and in particular horizontally acquired gene, because you can have access to available web tools like ResFinder. You see a screenshot of ResFinder with a result for 2 strains, one with a complete ICE and the other with a deleted form of the complete ICE. and you have a list of resistant genes. That's quite easy. But for chromosomal point mutation, it's less easy, and you really have to focus on some particular genes encoding resistant to quinolone, nitrofurantes, or the one that encodes the restoration of polybixin B susceptibility. For AMR structure, bearing this gene, it's more difficult, like the S60 element, the GI15, the plasmids, because you have to do blast comparison with reference sequence, and often you will have to perform long read sequencing. But at the end, you need to be a bit of expert of these AMR genes, because even with this tool, you can come across false AMR genes like KATB9, which does not encode chloramphenicol resistance. You can have a list of mutations that are actually not for point mutation, not associated with AMR. And also the first thing you have to do is to ensure that your genomic sequence does not contain contamination by other species. Next slide. So where are we now? Now, most of the isolates, that's a global phylogenetic tree here, are located in the red square. So you are at the bottom part of the wave tree. You are all the strains, they contain the variant CTXB7 of the base unit of the cholera toxin. And this clade was responsible for two major outbreaks, the one from Haiti and Yemen. Other researchers also found that these clade produce more cholera toxin, colonize more infant mice, and also more lethality to infant mice. So it's a more virulent sublineage of Vibrio cholerae we are dealing with right now. Next slide. And more virulent, but also more drug resistance, because thanks to genomics, we were able to identify and to follow a highly drug-resistant clone named AFR13 that was first seen in Yemen in late 2018, then in South Sudan, South Lebanon. I think that Antoine will speak about that later. Kenya and Uganda, so Eastern Africa now. So it's the first time we see such a diffusion of a highly drug-resistant clone, which is resistant to two of the three antibiotics recommended for the oral treatment of cholera, and even the bug can produce an ESBL. So that's the power of genomics, is to identify and to follow this kind of threatening population. Last slide now. So it's clear that microbial genomics has revolutionized, no, just before, the study of the cholera agent. But now, how can we use it for surveillance? So it will not replace a traditional microbiology method, first because it's too expensive, but so how can we use it in complement and in a synergistic way with traditional lab methods, culture or non-cultural methods? So I see three points, but it's open to discussion. First, we have to sequence Vibrio cholerae 01 CTX plus strains in non-endemic countries, and in particular in countries that are high risk for cholera introduction. Second, that would be very interesting to sequence a representative sample of Vibrio cholerae 01 strains per outbreak in epidemic countries for transborder surveillance, and why not establishing regional continental biobanks with representative strain for each outbreak. We could perform phenotypic testing also, and that would help us to identify, for example, a new resistance mechanism, and then to update our genomic tools afterwards. And last part is we can sequence over the course of an outbreak in case of change. If the bug, like in Yemen, has a new resistant pattern, if there is a difference in attack rates, in etc., we can sequence some strains, and maybe we'll see a second epidemic strain or an AMR acquisition or virulence acquisition by the initial strain. So that's some ideas that we can discuss later. And for me, it's all. Last slide. I would like to thank you very much for your attention, to thank all our collaborators. Next slide, please. And don't hesitate to contact me if you need help with your genomes. Thank you.
Thank you so much, Francois, for a great overview of a real global perspective of how genomics contributes to elucidating the epidemiology of cholera. So without further ado, we'll move on to our next speaker, who's Dr. Abu Fayed, who's an associate professor of microbiology at the Department of Experimental Pathology, Immunology, and Microbiology at the Faculty of Medicine at the American University of Beirut in Lebanon. His lab is also a member of the Center for Infectious Disease Research and a member of the WHO Collaborating Center for Research and Reference on Bacterial Pathogens. His main research focus is on antimicrobial resistance, with an emphasis on AMR in conflict regions. Dr. Abu Fayed is also an expert trainer in cholera diagnostics for the WHO cholera team in HQ. Antoine, over to you.
Thank you very much. I will be sharing my screen in just a second. Right. I hope you can all see my screen.
Perfect.
All right. First of all, thank you for the organizers for this opportunity, and I'm very happy to be talking to you today about our recent publication that we worked on in collaboration with various stakeholders, including Institut Pasteur and François Xavier's team, WHO, HQ, EMRO, and WCO Lebanon, the Ministry of Health in Lebanon, and the Lebanese University in the north of Lebanon as well. So to start with, since I don't have to introduce cholera because François Xavier already did this, I will be introducing Lebanon to the people, to those of you who don't know. Lebanon is a country situated on the Mediterranean coast. It's bordered on the west coast by the Mediterranean Sea, and then on the north and the east by Syria, and the south is bordered by the occupied territories of Palestine. And the region, Lebanon and Syria, and the occupied territories of Palestine have been going through some rough times for quite a few years, and has made it prone for cholera outbreaks to actually occur. We have a very nice coast and very nice snowy mountains, and we claim that you can ski and then go down to swim on the same day, especially in the months of April, if you're a good and fast driver. So, jumping onto cholera, October 3rd, 2022, we received a sample in the lab, and the Ministry of Health asked us if we can test it for cholera. The next day, we actually confirmed by culture that this was a Vibrio cholera 01 serotype Ogawa, and we then did antimicrobial susceptibility. We got the antibiogram, and we started receiving more and more cases as time passed, as you can see from the diagram here. However, an interesting turn happened on October 22nd when we received a different sample from a different area in the country towards the south. While that sample had a very different antibiogram of what we have been observing, and it looked more like the XDR strain of Vibrio cholera. So here it got a bit interesting because did the strain pick up a plasmid halfway through the outbreak, but this was still too early, so it didn't make too much sense. How did this resistance evolve and how did it emerge? So we decided to do some sequencing during the outbreak. But to summarize to you in a nutshell what we really did during the whole outbreak, we actually recovered 671 different Vibrio cholerae 01 samples from various clinical samples, and we've even received water and food samples that later on we discovered that testing them was not recommended. We've performed AST, antimicrobial susceptibility, on all of those isolates, and we found that 95% of these isolates, or 636 samples, had the what we called in our publication the AMR profile, while 35 isolates have had the XDR profile. And these 35 isolates came from very specific areas in the country, from the south, specifically in Tyre, and over at the Bekaa Valley. We've done a PCR, conventional PCR, to confirm the presence of the cytotoxin gene, of the toxin gene CTX, and we then chose 34 isolates that we've done sequencing for based on their various AMR profiles. Some of the isolates were sequenced in-house at AUB, and other isolates that were isolated by the Lebanese University in the north of the country were sequenced in François Xavier's lab in Institut Pasteur. So just to put what I said to you in context of the map, you can see within the map of Lebanon, only the white districts were cholera-free during the outbreak, while cholera affected more or less the entire country. Please keep in mind that during that time, as it is still the current situation, Lebanon has been housing more than 1 million Syrian refugees. It has the highest number of refugees per capita, and this also was one of the main drivers for the spread of the cholera outbreak. You can see the numbers here of the isolates that we chose to sequence. The yellow ones are the AMR profile, and the pinkish ones are the XDR profile. And you can see that they come within the Bekaa Valley here in the Baalbek Hermel area, and in the south towards Tyre. So after we did the sequencing, we sat down and we did some analysis. And as François Xavier mentioned, I'm not gonna dwell on it more. They belong to the 7th lineage. They all belong to the 3rd wave, and they're both of the sequence type 69. Sorry. However, when you look at their susceptibility profile, there is a striking difference when it comes to that. The more resistant strain had additional resistance to cefotaxime, to azithromycin and erythromycin, nalidixic acid, just like the dominant strain, to the 0129 as well, to trimethoprim sulfamethoxazole, and to nitrofurantoin. So what was quite interesting in the work we did was seeing 2 different introductions, and not an isolate that actually picked up a plasmid in the outbreak like what happened in Yemen. And you can see here with the phylogenetic tree that François Xavier generated, you could see that our isolates cluster well with isolates from Pakistan, which would make sense. This is the dominant strain that traveled from Pakistan to Afghanistan, then to Iran, Iraq, Syria, then to Lebanon. However, we've had a separate introduction for the Yemeni strain due to some political reasons in the country. The Yemeni strain, as mentioned by François Xavier, has this plasmid which harbors additional antimicrobial resistance genes. And you can see here with the circular plasmid, there is a good alignment between the sample from Lebanon and the sample from Yemen as well. And the AMR genes here are visible on the outside circle, where you can see the SOL1 gene, which is responsible for the trimethoprim sulfamethoxazole. We've got a PER7 gene as well, in addition to others. So an additional antimicrobial resistance database that in addition to the one that François Xavier showed, which was ResFinder, we've also used CARD, which we find quite useful. However, we could directly align all what we saw phenotypically with the genes present. So in other words, all the antibiotics that the bacteria was resistant to, we could identify the genes for. However, when you go the other way around, when you go from genotype to phenotype, when you look at all the genes, it's not the case where all the genes present correspond to all the antibiotics that are resistant. So if you would look here, for example, at the first line, the Vibrio cholerae contains the VARG gene, which is responsible for carbapenem resistance. However, we know quite well that the strains are carbapenem sensitive. So there is a SNP in the VARG gene which makes it inactive, keeping carbapenem sensitivity. But if you don't know or don't have the knowledge for this, you probably would fall for the mistake. So having not doing antibiograms basically would have misled us during the outbreak. So as a overall opinion and take-home message from sequencing cholera during an outbreak, I would like to add to what Francois Xavier beautifully said, which is sequencing is a very powerful tool for surveillance, and it gives us a lot of information, and quite a lot of times it actually gives us more information than what we know what to do with, especially if you would end up using both platforms, the short reads and the long reads, to get the circular genomes like we did in a good few of our samples. Basically, sequencing would never change and did not change our response to the outbreak in Lebanon. The response to the outbreak remained unchanged, and it did not really have that much of an impact on public health. Confirmation by culture was still necessary, and specifically antimicrobial susceptibility was very much needed for better understanding of the profiles and to recommend which treatment to use. So sequencing is a very useful tool to understand the surveillance and the molecular epidemiology. And for us, it served as a useful tool to understand the introduction of the second strain. It also led us to publish a very nice paper in Nature Communications that I invite you all to have a look at and read. Very powerful tool sequencing is, but does it really change the need for culture during an outbreak response? We do not believe so. At the end, I would like to thank all the collaborators and funders and the co-workers for their support, and I'm happy to answer questions when the time comes. Thank you.
Thank you, Antoine, for a really interesting perspective on a complex outbreak situation in Lebanon. So moving on to our final speaker, who's Gerald Mbar. He's a bioinformatics scientist at the Broad Institute of MIT and Harvard in the USA, and has extensive experience in advancing genomics-driven public health programs. He specializes in utilizing next-generation sequencing technologies to enhance infectious disease surveillance. Gerald is passionate about applying genome sequencing to health and disease research, outbreak investigations, pathogen characterization, and AMR surveillance. His work focuses on bridging the gap between innovative research and impactful public health interventions. So over to you, Gerald. Thank you.
Thank you very much, Tim. My name is Gerald Mbowa, as introduced by Tim, and I wish to thank my previous presenters, Anton and Francois, for laying the ground for what I'm about to present. So I'm presenting work that has been done through Africa, by Africa CDC to ensure there is enhanced cholera genomic surveillance in Africa. So a brief background, I know it has been covered extensively, but this is an outline of what the outbreaks have been in the past years. And recently, more important in Africa, where it is endemic, there have been multiple outbreaks that involve endemic and non-endemic countries, as we shall see. And they have been characterized by high case fatality rate over the years.
Gerald, just to interrupt, so your slides aren't in presenter mode and they're not going forward.
Or can I request to, so they are shared from your side? I think it is a problem on my side.
No problem.
Oh, okay. Sorry about that.
Sabria, can we share Gerald's presentation, please?
Sorry about that. Can I try again and see if Supriya is unable to share them?
Yeah, please go ahead, Gerald.
How about now? Okay, so this is what was in the news during the previous outbreaks. We had the outbreak from Zambia, where we had a death toll hitting over 700 in the outbreak. And then later on, we had some news reports that were looking for what was fueling this outbreak in Southern Africa. And then later on, we had scientists arguing the possibility of doing genomic surveillance, potentially looking at new variants that were responsible for the outbreak. So what came in was Africa CDC responded by bringing together different collaborators and international partners to establish a consortium called Pathogen that specifically looked at understanding the genomics for these outbreaks. And
sorry to interrupt again, Gerald. They're still not showing, so I will share from my screen. That's okay.
Okay, please do so.
It's okay. I'll share it, Tim.
Okay.
This is
Right.
Supriya.
Sorry about that.
Can you see it now?
Yeah, we can see. Can you have it in slide view?
Yeah. Which slide is it?
You can
Tell me
go.
when to stop.
I'll tell you. The next slide. Yeah, so the next slide is here we look at what Africa CDC formed the consortium that was basically going to analyze the genomics of the outbreaks itself, try to understand the transmission pathway, try to look at antimicrobial resistance patterns that could inform vaccination strategies, but also leveraging the regional genomic capacities, and also understand the challenges and how this could affect policy, and also understand the cross-border transmission across the affected countries. Next. So this is the consortium that we named Pathogen that brought partners across United States, that is Johns Hopkins University, to provide the genomic expertise and epidemiological aspects of this consortium. And then we assembled countries across the affected areas, but also countries that were endemic to cholera, that is Nigeria and Cameroon. And what you can clearly see that each of these countries share country borders. So you have Nigeria and Cameroon, then you have DRC and Uganda and Zambia and Mozambique and Malawi. So you clearly see this belt has countries that were endemic to cholera, but also countries that were newly affected by the outbreak. So that was a consortium that was assembled, bringing 7 countries in total in Africa, and then coordinated this response through Africa CDC. And what we are looking at under Pathogen is to provide answers to the questions we have seen above, whether this was a new variant and whether there was changes in the antimicrobial resistance patterns or profiles across these affected regions, because we are reporting high fatality rate and outbreaks across countries like Zambia, where in more than 20,000 cases, Mozambique, more than 40,000 cases. This had not been reported before, especially these new areas in Southern Africa. So we basically also used genomic surveillance to explore the diversity, identify new lineages and microbial resistance patterns, AMR profiles, and also to try to understand the direction of cross-border transmissions. Next. Yeah, so the consortium, like I've already said, working and coordinated by Africa CDC and international collaborators, established this genomic surveillance network, Cholera, leveraged local sequencing capacity that had been built during the COVID-19 pandemic in Africa. And we managed at the moment over 1,000 genomes have been sequenced across 7 countries. And some of them are from the previous outbreaks across the different countries in Cholera, but also we are lucky to have countries that we are newly reporting cases, that is for Zambia, Malawi, and Mozambique. So that added a new component to this genomic and surveillance that we carried on. And then the objectives were basically to build capacity for bacterial pathogen genomic surveillance in Africa and bioinformatics using specifically cholera, which was a priority pathogen of interest in Africa, and then also determine the origins and reservoirs, the genomic diversity, and the transmission patterns across the cholera countries, but also across the border. Next. So we had our first training last year in September at INRB in DRC, where we carried out, we trained the different laboratories that were participating in collagen in genomics sequencing of Vibrio cholera. So what happened was the countries identified our isolates from their biobanks across 5 years, that is starting 2019, because as you saw from Francois' presentation, there is a study that looked at over 631 isolates from Africa, and those were up to 2019. So we tried to leverage a gap that had not had any sequencing of any isolates from Africa, starting from 2019. What we know during that time, the pandemic, COVID-19 was the biggest concern for most of these countries. So during those outbreaks, isolates starting 2019 were not so much represented in the genomic sequence databases. So these countries came with hundreds of some of these isolates from their country borders, and then we tried to sequence them at INRB. So that was the training that was used to build capacity for genomic sequencing of Vibrio cholera. So they went back into their countries and they continued doing sequencing, sampling again through different outbreaks over those 5 years. Next. So the challenge we got here was the bioinformatics analysis. Following the sequencing itself, you clearly see we're struggling to have each individual run through the analysis of their samples, their own samples. And what we know here, our plan was to sequence close to 1,000 samples. So you clearly see we're struggling to see how best we can coordinate 7 countries in one place, running through the analysis of their own samples, but also the samples from other countries. So this was kind of the situation we found ourselves in, and we're trying to do our best to ensure that we can sequence those samples in a way that we can offer insights for each of these countries. Next. So we used the Terra platform, which is more of a collaborative platform that offered us a kind of an ecosystem where each of these countries could remotely go back to their institutions, upload these samples within the platform, and then virtually we could coordinate the training. So we had the first in-person training at Addis Ababa at the Africa CDC headquarters, where we trained them on how to use the platform, so that when they went back to their own countries, they managed to upload these genomes within Terra, and then we are able to coordinate the genomic analysis of these isolates for each of these countries, but also giving us an opportunity to merge the entire data set of over 700 samples, isolates within the same ecosystem, and derive insights that informed us on how the transmission was occurring within the country borders, but also across the different countries within Cholera. Next. So this is what we managed to do. We confirmed the isolates that we managed over out of like the detection, I think, was slightly above 60%. So out of the 1,000 isolates we sequenced, we had over 728 that were confirmed as Vibrio cholera through genomic analysis. And also we managed to do the typing of those isolates. And then we did variant calling to try to understand the different variants and offer the annotations, phylogenetic analysis, AMR profile. We inferred also the transmission and epidemiological analysis,
Thank you, Gerald, for the excellent presentation and the really impressive work of Colgen of the network. So I'd just like to thank all of the speakers now and hand over to Nadia Vokia, who is the laboratory focal point for the WHO cholera team in Geneva and for the Global Task Force on Cholera, to guide us through a short Q&A session and then provide some closing remarks. So over to you, Nadia.
Can you remind us how long we have now for Q&A? Because I think we're
maybe five minutes or so. Maybe a couple of good questions.
All right. Thank you very much. So yes, so there are many questions in the Q&A. So I do want to ask our kind presenters to go and have a look at those and answer as many as possible. And there are some hands up, and they have been for a while. So I'll go to the hands if that's okay for now. Maybe the first one I see, Sophie Devos, you have a question for our panelists. Please go ahead.
Nadia, could you please read out the question, because we don't have the ability for panelists to speak.
Okay. All right. Apologies for that. All right, then I'll go into some of these questions. We had Flavio Finger that was asking, this was on François Xavier's presentation. He says, thank you very much for this interesting presentation. What do these results that you've presented mean for the widely quoted theory of environmental reservoirs of cholera? Would you kindly comment on that?
No, 5 minutes is not enough for that. No.
We can go a bit over, it's fine.
If you want to sample environment months, years after an outbreak, when it ended, you will never find the 7th pandemic cholera agent. You might find other Vibrio cholerae, but not the one that was responsible for the outbreak in between two outbreaks. No. I know people that did a postdoc in a famous environmental lab in the US, and he tried for years in Senegal after the last outbreak, and he never found the 7th pandemic cholera agent. Other Vibrio, yes, but before the confusion was between these pandemic lineage and other Vibrio cholerae. Now we have the clue to differentiate them, and there is no long-term aquatic reservoir for this pathogen. You can find it in weeks after by fecal pollution, but not as a perennial aquatic reservoir.
Thank you very much for that. There was actually another question in these questions. What about the other Vibrios, right? Vibrio parahemolyticus, vinificus, and their power of potentially creating outbreaks and genomics. Do you want a quick word on those, on other Vibrios?
It's not the topic for this. We might have another session dedicated to this. It's different epidemiology. Of course, in this case, there is a link with the sea, but it's a different story.
Understood. Okay, we'll keep that for another time then. Okay, next question. I think it's also for you, François-Xavier. Is genomic AMR testing applied clinically for antibiotic selection?
If it's antimicrobial susceptibility testing is performed with no good standard, then that is a method of choice. Genomics is just a way to be more discriminatory, to find the genes, to understand the global evolution, but no. But sometimes it can be helpful because when you test with disk that you are not sure, maybe ill conserved, you will see an inhibition zone. The categorization can be different with just 1 millimeter difference. Then genomic can be helpful because the gene is here or not here. But no, if you're antimicrobial, that's the gold standard. It's antimicrobial susceptibility testing by phenotypic methods to guide clinical treatment. Genomic is just at some point to have a higher view on what is going on, but not to guide treatment. I hope Antoine is also okay with that.
Absolutely.
Excellent. Thank you both. Another question for the panel, whoever wishes to respond. Do the genomics results, can they be, the question is in French, sorry, so I'm trying to translate in my mind. What of the genomics results today and climatic change? Do you have any comments on that topic? Quite a broad question again.
Climatic change can lead to droughts, floods, and leading to human gathering. So it's maybe an indirect impact on cholera than rather a direct. And genomics, big question.
Understood. Dr. Gerald, there are some quite a few questions for you too, so please do go and look in the Q&A and respond when you can. Questions such as, there's a question on why South Africa and Zimbabwe were not participants in the Cholera given recent outbreaks. Also, are there efforts to review the genetic sequencing and AMR patterns in Nigeria? The speaker adds, the person adds, there are persistent cholera outbreaks in northern Nigeria, especially Kano State. Could you comment about those countries if you have any information?
Thank you very much. And what I can say is for Nigeria, our Cholera continues until next year, 2020, almost early 2026. So we continue to do prospective and retrospective sampling and sequencing. Why South Africa wasn't part, I think there's a lot that had been done when it comes to capacity building. So we are building capacity in areas that were either having an ongoing outbreak and didn't have the genomic capacity, of which South Africa wasn't, and also Zimbabwe didn't have so much to the scale that we saw in Zambia and Mozambique. So I think those were the reasons as to why they are not part of the consortium.
Understood. Thank you very much. Do we have any comments about, so the question is framed as this percentage of XDR in Vibrio cholerae, but I guess so understanding of how widespread in terms of numbers as well. I think François-Xavier, you did touch on this during your presentation.
Yes, we are monitoring the spread of an XDR clone, but we don't have figures. For example, what could be done is that when countries report cholera case to the WHO, they do not report the characteristic of the strains. So for our strain, we got the strain through a surveillance of travelers in Europe. When I mentioned Kenya, it was 3 travelers from the UK, France, and Belgium that got ill in Nairobi in 2023. And when you look at the data to the Kenya reported case, but we don't know the number of cases with this strain and another different resistance pattern. So it's impossible. So maybe we can, we could in the future report case and the characteristic of the outbreak strains. That could be helpful to follow XDR, because it seems to just to be starting now.
Understood. Yeah, you raise the important question of the sharing of data and how a lot of this information cannot be done without all of this data being made public in some way, shape, or form. We definitely support that. Thank you very much. There's also a question about the vaccine, right? Cholera OCV and genomics. So how does these two, go in parallel? What do we know today? Have there been any changes, etc.? So the study of genome of cholera, has it had any impact on OCV? Could be any one of you.
I can,
All right.
I
Yep,
can
go ahead.
jump in on that if you want. So we know for a fact that genomics aided in COVID vaccination development. It is aiding in influenza vaccination development. But if we notice that these are still viral diseases, right? We haven't yet touched base on bacterial diseases. That's point number one. We would see at some point eventually the role of genomics when it comes to bacterial diseases, vaccine development. We no doubt will reach that point. But at the moment, the good thing is to collect genomic data when the time allows, not use genomics for cholera to respond to an outbreak, but to do active surveillance and to actually understand the spread and the epidemiology, the molecular epidemiology of what's circulating, and collect that data to be able at some point later on to help with the development of new vaccines if needed. However, the current vaccine, the OCV vaccine that's currently present, actually covers all strains circulating and has no issue whether there is more AMR or less AMR within the strain that's circulating.
Thank you very much, Antoine, for these clarifications. And maybe finally, there was a question for you, Antoine. Oh, I think you answered it, about the political reasons for the outbreak in Lebanon, right?
Yeah, I answered in the chat. I put that in the chat.
Thank you very much. I know I've missed many questions, but Supriya and Tim, I think we've touched on quite a few important points there. It would be nice to have another hour to discuss these. Would you like me to continue?
I think, Nadia, we can have your closing remarks now.
Okay, very good. Well, first of all, let me extend, my sincere thanks and from many of us here for all these great talks and to all the presenters. Very, very comprehensive and very interesting, and we look forward to continuing these discussions in the near future. These presentations have underscored the critical role for genomics in cholera surveillance, definitely touching on many of those aspects. They particularly illustrate how genomic insights enhance our understanding of cholera's global circulation, right? With the notable recent focus on Africa, and we're looking forward to see more results there too. We're very pleased to note that what was once, limited to international laboratories is now becoming accessible to pretty much all. This is due for different reasons, right? Simplified sequencing methods, localized implementation of the necessary tools and competency, reduced costs, making the technology more affordable. So this aligns very well with what the Global Task Force on Cholera Control's objectives to support real-time surveillance of cholera transmission. However, I think as a conclusion, we do want to highlight and really emphasize that genomics in the context of cholera serves for specific, it serves a specific contextual role, right, in surveillance based on the epidemiological context. It's not sequencing for any purpose and all the time. We have a lot of work to do within the GTFCC to continue to clarify those questions and really help these activities moving forward, I believe, personally. As highlighted by the GTFCC surveillance and laboratory working groups, genomics is not intended for primary identification of Vibrio cholerae. In fact, we find that it's even a disservice to the powerful, to these powerful tools to think of it in that way and to use sequencing to simply identify VC or serogroup, serotype, etc. So we're looking forward to more efforts in really shaping those questions moving forward, those well-defined questions. Thank you very much once again. I'll stop here. And yeah, enjoy the rest of your day.
Thank you very much, Nadia, and thank you to Tim for moderating the whole webinar, and of course to the speakers, Professor Francois, to Antoine, to Jérôme, and most of all to all the participants. I can see all the emojis coming up now. I can see we have about 50 questions and lots of comments on the chat. So thank you very much to very engaged participants. It's been a pleasure to have everyone over, because this is the last and final EPIWIN webinar of 2024, and this year has been fantastic. Thanks to demand from all of you, we've had 30 EPIWIN webinars. We were originally planning on 20, so that's 10 more than we expected. We hope to continue to meet all of you next year. We will be starting again in the second half of January, so watch the space. But in the meantime, have a wonderful holiday season for those of you who celebrate Christmas. Wishing you a very happy Christmas and a wonderful 2025. See you next year. And thank you from the EPIWIN team. Bye.
Thanks all. Bye-bye.
Thank you. Bye-bye.